And hey everybody, Wayne and Mark B on the behind the screen, and you can hear the birds tweet when he says hi. So Mark's in Wisconsin and Wayne's in Colorado and Mark's in Bangladesh. And that's about it for announcements. I'll turn it over to Mark and let him go at it.
And away we go. All right, as you guys can tell, we're approaching the end of our forest ecology series here, and that we've gone from the big picture and general concepts of ecology in general, and now we've gotten to the point where we have our basic homework assignment where we're going to do a restoration ag mimicry of a natural plant community, and then now we're getting into some of the finer details at the organism level, and this week we're going to deal a lot with heredity and variation within plants. And you guys know me well enough. I can't stop trying to make this relevant and practical on the ground to what it is that we do, as restoration ag practitioners. And so that'll be all through here. And got to figure out how to run the screen again. There we go. There we go. And as you guys have heard before, this is a book right here that basically changed the course of my career as a student, and then of course my professional career after that. And when I read Tree Crops, originally penned in 1926, A Permanent Agriculture. That's the first time I ever put those two words together, permanent and agriculture. I'm reviewing this book right now, and it's amazing how many of the different things in Tree Crops, the whole outline of this whole course, you can basically find it in Tree Crops. All of what he proposes to come up with a new type of agriculture is all of what we've been talking about for these past 30 some odd weeks. So hats off to J. Russell Smith. And of course, Bill Mollison, yes you may.
Can I jump for a second before you leave Tree Crops? Are you at all able to give a little bit of an insight of what might be happening with Tree Crops?
Well, Tree Crops, the copyrights have long since expired, of course. The last publisher to actually deal with it was this one right here. I believe it was Island Press. And what has happened is Acres USA has contacted all the previous publishers, and including Wendell Berry, who did the introduction to the Island Press version in 1986. I think it was 1985 or 6 when it came out. And Acres will be putting out a new Tree Crops, an annotated and updated version of Tree Crops. And the person to do the annotation and updating will be me, yours truly. And so currently what I'm doing is I'm reviewing the book and going through it and finding places that deserve some additional commentary, because it's been almost 100 years since he wrote this book. Some things we've learned better than what he said. Some things, we're not even close to what he had imagined. And so I'm in the process right now of making notes of all of where that's going to be. And hopefully, I don't know if it's going to exactly happen, hopefully this will be published, released at the same time next December at the Acres USA conference, at the same time as the water management book. So that's a little sneak peek of what I get to do. Man, I don't know if you guys have ever done any writing to do it as like an occupation. I'm like a pick it up and put it down kind of guy, put the trees in the ground, hop on the tractor, cut this, drag that. To me, that's work. To sit down at a computer and read a book and type is just like frustrating. You look up and 3 hours go by and you haven't done anything. So it's a little frustrating, but I'm having a lot of fun reading through the book again.
Well, I'm going to throw two other things out there, both cool. By the way, you're getting lots of comments saying things. One is, I think I also heard that Wendell Berry might do another introduction, correct? Potentially, at least.
His daughter, I guess, has said that either
his daughter,
will,
right.
But his daughter said that either he will or she will. And so if he doesn't, that'd be fine if she does it too, because here's a new, I'm a young generation compared to Wendell Berry, obviously young generation compared to J. Russell Smith. So it'll be the younger generation kind of picks up the work and takes it forward.
And then one other thing related, I think you've said, and this is, please don't take this, guys, as absolutely what had happened, but part of what might come from these assignments that you guys are all working on Might have some relevance with the book, right, Mark? So you might just mention that.
Not necessarily with the tree crops book, but it absolutely will have some relevance because that also is being talked about with Acres USA. All of these webinars are going to be summarized. They've obviously been recorded, going to be edited down to get down to the meat, and then transcribed, and then flesh out the transcription, you know, because it's happening tonight already again. I'm trying to move through a slide, and we start conversation here, and we do conversation drift. But when we have it all transcribed, I'll be able to fill in and have it in an orderly outlined fashion. So there will be a book coming from this, and all of the people who do one of the assignments, that will be included in that book when that comes out, with proper credit to you as well.
And that's kind of what I meant. So anyway, let's keep going. Sorry.
Go flash through this, and part of the whole aim of whether it's J. Russell Smith's type or, you know, the permaculture type, the aim is to create systems that are ecologically sound and economically viable. How can we design a system that's ecologically sound unless we know how ecological systems work? That's what this course is all about. And the economically viable, that's something that we're all working on. And one of the things that an ecological system helps us to do is to lower our expenses to approach the level of what nature does on actual monetary expenses. And nature doesn't spend any money on maintaining ecosystems. So the closer we get to being a real natural ecological system, the less expensive it's going to be to operate. That said, a natural system doesn't necessarily maximize yields. Well, we don't necessarily want to maximize, you know, one particular yield either, but we would like to optimize production. And so we have a lot of the tricks that we'll talk about, a lot of the general principles that we'll talk about tonight are directly applicable to how we get increased yields in our systems through time. So what restoration agriculture is redesigning agriculture in nature's image. We're going to be doing biomimicry. We're imitating nature, doing broadacre permaculture, whatever you want to call it. I wrote a book in case you hadn't known that. acresusa.com, go buy it. And so basically what we're going to do is identify our biome, and within that biome, our dominant plant communities. We're going to optimize the rainfall distribution pattern to prevent runoff, et cetera, and to establish aquatic systems, which have synergies for pest control and fertility, additional food for people and animals. Then we'll establish woody polycultures just like nature does, but we'll use the agroforestry techniques to get there, and then we'll manage that system for as long as it goes. Now, we've seen this slide, I don't know how many different dozens of times here. This is what was here in North America in these particular regions, also existed on the West Coast. This plant community, basically the oak savanna plant community with oaks, apples, hazelnuts, plums, cherries, peach, raspberries, grapes, currants, pastures, animals, and fungi. This is a natural system. Of course, before people got here, the species were different. What we can do now is substitute our chosen species for these and manage it as a farm by harvesting yields. We harvest the grass using animals. We, increase the soil fertility using the animals, crop rotation, appropriate cutting, chipping, et cetera, et cetera. Well, what's fascinating about this, how is it that oaks were able to survive way up here in Manitoba and way down here in Texas, what the heck, all the way down to Mexico, into Mexico? There's an amazing variability within the oak itself. Same with the apples, the hazelnuts, the raspberries, grapes, etc. There's grapes that'll survive in maritime 60, 70 degree rain in coastal Maine, and there's grapes that will survive out in Texas, almost the desert, for crying out loud. This ability to survive in different environments is genetically driven, and that's the genetics of plants is a very useful tool. This oak plant community thrives here. That's coastal California. It's probably now green right now or landslided, and it also thrives here. You'll find the same plants, but just different variations of them. And all plants, they need some sort of mechanisms internally that allows them to pass their behavior, their traits down from generation to generation. And the thing with trees, plants, trees especially, is they're even more critically dependent on their environment than we are. If things get rough for us right now, we're all going to move to Panama or the Dominican Republic instead of staying in the USA. Well, a tree just can't pack up and go when the resources change or the weather changes or the climate changes. It's stuck there sometimes for thousands of years. Its form, the shape of the tree, how it holds its branching pattern, whether it's straight and tall, whether it's open and round, the form, the physical form of the tree is a beautiful blending of its genetics and its environment, because the environment is what kind of triggered certain genetics to turn it on, shut it off, and the plant responded by behaving a certain way, responds to environmental stimuli a certain way. So that appearance of that plant is the phenotype. And both the genetics and environment combine. It's not an either or. Here's a classic example. Is a tree like this on the right. This has been growing amongst others in close competition for light, and it could not spread out to get any light. It had to go up, 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 up. And the lower branches get shaded out and they self-prune, they fall to the ground. And so this tree on the right and this tree on the left, you can take a clone or a rooted cutting of the same exact tree and plant one in a thicket of other trees. It has to grow straight up, like on the right. Or if you plant it out in the wide open, it has all the sun that it wants. It can spread out and tries to capture as much sunlight as it possibly can. The actual mass of the tree might actually be the same, but this is in a wider spread branching pattern, and this would concentrate more on a long, slender trunk. That's a combination of the genetics and the environment. This particularly right here shows more the environmental effects on that particular tree. So whenever you see trees like this, most likely they were open grown out in full sun. And anytime you see trees with long trunk with no branches on it down low, it's grown amongst lots of competition. And we'll see some examples of that tonight. So the phenotype, the appearance of that tree, can assume different characteristics in one environment versus another. And its ability to change its form depending on the site conditions is the plasticity of that phenotype. And what I showed you in the previous slide, that shows you exactly right here. If this is white oak, for example, this shows you the plasticity of that particular phenotype. It can respond by doing this, or it can respond by doing that, or any number of forms in between. It's a very plastic phenotype. Another plastic phenotypic appearance that we don't necessarily see all the time is root form and root growth and the root depth. A lot of plants, if you have, let's just take hazelnuts because I plant a lot of them. If you plant a tiny little hazelnut plant in a heavy clay soil, and it's been growing in a little pot, a little tube, and it hits the outside of its walls, the roots send a signal to the plant, say, hey, yo, whoa, we're stuck. There's plastic on the outside. We can't go anywhere. Stop. So it builds a lot of fibrous internal roots. It'll hold the root ball together really nicely. But if it stays in that confined container long enough, when you take it out, it's already sent a signal to the plants like, oh, we have a small root system. We will never have a big root system, and it may never grow. One of the things to help it to wake up, if it hasn't gone too far, is to make sure that soil has a nice deep rip line in it with a subsoil or a yeoman's plow, just to get a really deep, loose, crumbly soil so those roots can run. Because the plants will oftentimes, they'll form their roots based on their conditions. So this is a phenotypic response to different site conditions at planting time and as they've grown. So the phenotype is the exterior appearance. The exterior appearance, of course, is a result of the environment interacting with its genetic makeup, its genotype. And here's the range of various different pines throughout North America here, and it's obvious. You can tell for sure that some lodgepole pines growing up in Yellowknife. Where's Yellowknife? Isn't it? Is it up in Yukon? I think it is. No, Yellowknife might be in Northwest Territories. Anyways, lodgepole pine growing up there is going to be a radically different genotype than lodgepole pine grown in Colorado because of the difference in the environment. If you are not capable of surviving, 100 million below zero for, months and months on end, and if you're not capable of surviving with this really strange photo period, we have long days in the summer, and then you get deadly cold before the days shorten up. The plants that don't have the genetic makeup to survive that, those genes drop out of the gene pool. Whereas in Colorado, it's still a lodgepole pine. The two can cross-pollinate, but it'll have a totally different set of genes inside that code for different strategies. Perhaps drought is one of the characteristics that it would have in it, or surviving heat, because it can get blazing hot in some of the southern ranges of lodgepole pine. So the genetic makeup is the genotype. And once again, it's the site conditions that determine which genetic traits, which genes inside the DNA of that tree are going to either turn on, certain environmental stimuli turn certain genes on and they'll start coding for certain proteins and certain enzymes, or other stimuli might silence those particular genes, and these genes won't express, and those are turned on and off by the environment. Those are epigenetic changes that take place in it. Well, then there's the reproductive genetic exchange between parent and child, where a new seed is formed, an embryo, the fertilized embryo, and then that new seed has a new scrambling of the genetics that's never been there before. And so the ability of the organism to live and reproduce in a given range of environments is adaptiveness. I chose this picture right here, and we'll see this a couple times, simply because white spruce is one of the more adaptive trees out there. There's white spruce, seriously 3 inches tall, surviving out in the tundra. There's white spruce surviving on a rock in Isle Royale National Park. That big, it could be 100 years, and it's that big growing on a rock. This kind of cold, this kind of wet. The tallest white spruce that I know of in the world, I think, is in Washington State. It's just a monstrous tree. I think that's the picture of it right there. So it's a rather adaptive. The genetics of white spruceness is very adaptive. You can throw it in all kinds of environments, and it's likely to have some sort of variant that will survive and thrive. And then, although all the cells of a plant may have the same genetic makeup, the internal environment of that plant also expresses what genes are expressed, what enzymes are produced, what characteristics the plant will have, and it's like the magic. I don't know what to call it besides the magic place is the meristem, the apical meristem. We've heard of stem cells when it comes to human research, but meristem is where that word comes from. It's meristematic tissue. It's undifferentiated tissue. And inside of a bud, there's a region of meristematic tissue that can turn into anything. And for some reason, that meristematic tissue will know when to send chemical signals to the rest of the plant saying, yeah, we're a juvenile plant. And like in the case of honey locust, it will grow with these massive thorns all over it until it reaches a certain age. And it says, we don't need thorns anymore. We can now turn into an adult honey locust. Those chemical signals take place and are originated right in this meristematic tissue right here at the tip of the bud. A cabbage is basically a gigantic, oversized bud, and this is this is what I call the bud signal. This is bud man right here, bada 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 bud man, and this is the magic in a plant. The cells in here are dividing incredibly fast. There's a lot of cell division going on. During cell division, I'll show a little bit later on, there's all kinds of crazy things can happen in there, from mutations to crossing over and so on. So every time you see a bud on a tree, potentially that has a new genetic combination in there. If you have a tree with 10 million buds, it is possible, I don't know that that's the case, but it is possible that every one of those buds has differentiated slightly differently and the genetics have changed, and you've got 10 million different separate genetic individuals on one tree. And they guide the growth and the form and the behavior of that particular branch. So mutations and recombinations. Mutations are when there's a a cellular division and the DNA or the RNA, ribosomal RNA, divides, and there is a change, an error in the code, in the code that is the DNA that tells how to make proteins or how to make enzymes, etc. And that's all a mutation is. Not all mutations are successful. There are, I guess, many mutations, if not most mutations, are benign. They don't seem to do anything because that particular trait isn't expressed in the phenotype. There are some, obviously, that are harmful. There are some that are actually beneficial to an organism. The recombination of genes, of course, is during cell division, during meiosis, when we make the different gametes, the male and the female gametes, pollen or the embryo in flowers and plants, that when they recombine, now you have some genetics from parent A, some genetics from parent B, and now the two come together into a new form that's not happened before. What also can happen here is at this particular cell division phase, you've got all the chromosomes have lined up across the equator here, and they're about to be pulled apart and go to their own separate pole. However, it's a messy process and they're all tangled together. Sometimes what can happen is this little leg over here can cross over that leg over there, and for some reason they just hook. And so you can take a piece of, one chromosome, moves completely over to another chromosome, and it has a different output. It will just make a different protein or a different enzyme, and that can turn into radically different downstream results. So there's mutations, recombination, and I didn't put that last one in there. That's crossing over. Crossing over also occurs during the cell division phase, and it's most critical. You think about that. It's most critical in meiosis when it's making the gametes, the male and female reproductive cells, and it's the most critical as soon as they come back together again, and then they start dividing to turn into the new organism. As soon as they start to divide and go into two new cells, any change that took place at this level right here is going to be propagated through that whole entire organism all the way through. So continuous variation is typical for most plants. And what that means is polygenic variation means that there's a lot of different genes, a lot of different parts of the chromosomes, and sometimes multiple chromosomes that end up being coding for a particular trait. It's not as simple as like with eye color in human beings, where it's a small little piece of DNA on the X or the Y chromosome that codes for your eye color. A lot of the different traits in a plant are on multiple sites on one piece of DNA and multiple pieces of DNA that all lead to certain phenotypic appearances and or behavioral. And I'm calling it behavior. I don't know if you can really call how a plant grows and moves, interacts with the environment behavior, but I guess that is for a plant. It's behavior. And so that causes challenges for people who want to do a very carefully controlled breeding with plants. And fortunately for us, we don't have to limit ourselves with single pollen grain controlled crosses. We're this. We like throwing that pollen around all over the place. Now, it's the exchange of genetic material between the different populations is the gene flow. Migrants from as a form of pollen or seed bring to a population new genetic material from other populations. So right here, here's some, this is actually, I believe it's limber pine, Pinus flexulis, that all this pollen is floating through the air. It could go up into the stratosphere and float around and go 3/4 of the way around the world and come down in South America someplace and land on a receptive flower of another pine tree, and it happens to make contact, boom, the seed grows. You've just brought new genetic material to that particular plant in South America. So this gene flow is part of what's critical for the whole population as a whole. And as we'll talk about later on, it's the populations and it's the communities in a larger scale that are what it's all about, not necessarily the individual plants within any system. the health of the system is dependent on the health of its overall gene pool, not the health of individuals within that system. And of course, we got squirrels here moving seed around. They may get picked up by a hawk and then eaten, and then the seed that the pine cone he had in his mouth gets dropped on the ground somewhere 20 miles from where it started from. Or these bison go on a huge migration from Oklahoma all the way up to the Dakotas, and some of the seeds that they carried on their forehead, now he scratches on the bark of a tree and the seed falls off and sprouts. So now we get a new individual growing there that then starts to cast pollen and receive pollen. So we've now added new genes to the gene pool by transport from animals, wind, etc. Now, when species are substantially different, I had mentioned a couple slides ago that pollen, the Flexilus, Pinus flexilus, limber pine, it could land on a lodgepole pine, and it may or may not pollinate that lodgepole pine because they're not closely related. They're closely related, but not extremely close. And let's just take an example right here. When you have two populations that are substantially different, they're different at the species level. That process is termed hybridization. Hybridization gets a lot of bad press because the big, nasty, evil seed corporations have parent A and parent B. You get the offspring, which is a cross between A and B. And if you take all of your offsprings, which are the F1 generation of parent A and parent B, and you cross them together, your likelihood of getting the same thing that you planted is now at least 16 to 1 against you. And you'll have to rebreed to get back to a stable population, no less than 4 generations to get back to stable genetics. And if you're the seed company, and let's take table ace acorn squash. If you're planting table ace acorn squash, it's recognizable in the marketplace. If you save your seed from that, you can get all kinds of wacky little variants if you save your seed. Whereas if you want to get something that looks like a table ace acorn squash for that market over and over again, you got to go right back to the seed company. So that's how it's used as a profit-making center for those seed companies. I do a lot with hybridization. It's all open pollinated. They do it all by themselves. And here's 2 different phenotypes. You look at this tree right here, straight as an arrow, really fast growing, shoots up, keeps its branches very upright. That's the American chestnut, Castanea dentata. And over here on the right is a Chinese chestnut, Castanea mollissima. It's genus and species, Castanea, Castania, Castanea, however you pronounce it. Canadian will probably pronounce it some of these different. So when you cross the species level, that's a species hybrids. So I'm doing a lot of work with species hybrid chestnuts. These are leaves from various different chestnuts, from Americans to Chinese to European to Japanese, and I'm not exactly sure what that one is right there. When you then combine them, you'll end up with all kinds of different variabilities in here. So what we're doing is we're intentionally introducing variability into the gene pool in order to create new varieties that have particular traits that we might want. What that means is not all reproductions are going to be favorable. There are going to be some that are not favorable. And the ones that are not favorable, so what? We cut them down, we inoculate with mushrooms, we put them in the wood stove, we make lumber out of it. So when doing breeding with plants, animals, the same thing falls as well. We look up over here. This is just shorthand for all of our levels of classification. It's kingdom, phylum, class, order, family, genus, species. Plant is known, animals are known by their genus, species. The genus name is capitalized. The species name is not capitalized. And when you put something in this binomial nomenclature here, it's supposed to be in italics. If you want to breed 2 plants that are the same genus, Malus, for example, Malus. Malus baccata, Siberian crabapple, and the Malus domestica, the European apple, that is very, very likely to be a successful cross because you're only crossing this species with that species. If you were to cross a Malus domestica with a Malus domestica, almost 100% success. So if you have like a Baldwin apple and a Red Delicious apple, they'll cross, piece of cake. You go one step removed from the species level or at the genus level, Malus to Malus, still very high success, but a little bit less than if you're just doing within genus and species. When you go to the genus level, it's a little bit less. You go to the family level, like down here with hawthorns and apples, they're still in the same family, in the Rosaceae family, but they're just enough farther step away that it's going to be a lot less likelihood. So it's almost geometrical that every step you take, it's geometrically less likely that you're going to be able to cross the two together. So a hawthorn crossed with a domestic apple, yeah, maybe it's possible. You can do that, and why not try, right? Now, when we're talking about plant communities, the different woody species, perennial species we're using on a farm, the individual is not the unit. It's the whole interbreeding population. So we've got to think now of our trees, if you want to call it an orchard, I don't want to use that name anymore. We have to think of it as a herd. We're looking at this whole herd of trees. And it's the herd that's important, not the individual trees within that herd, although yes, they are kind of important, but it's the overall health of the whole herd that we're looking for. That is what that is what has the strength and the resiliency, and that's the gene pool that we're working with, and it's the gene pool of the herd of our trees that have these heritable variation in the genotype. Now we can manage our selection, and the site will actually do natural selection for us through time. And natural selection is just when the site conditions are such that this particular phenotypic variation can't survive there because it's too cold, too wet, too hot, too dry, too acid, too alkaline, whatever it is, and that genetic variant just can't survive. So in the case of a tree, it will either die, or we see that it's struggling, and we'll cut it down and use it for some other purpose. So the numbers that we're talking about with trees is almost astronomical. 1959, our friend from UW-Madison, John Curtis, counted. What kind of a crazy guy counts 6,678,400 sugar maple seeds from one tree? That's insane. And then you go germinate them. This guy was obviously needed some more fiber in his diet or something. Only 57%, 55% germinated. That was 3,673,100 seedlings. By the end of that summer, only 198,000 seedlings remained. 2 years later, that's down to 35,000. It was less than 0.1% survived 2 years. Now think about that. Of all the different seeds of that sugar maple, less than 1% survived 2 years. So what was the biggest killing time period for trees is in their youth, is when they're very, very juvenile, when they're young. And that's when the environment, the environmental tests, once again, the soil, tilth, the heaviness, clay, sand, acidity, alkalinity, or percent organic matter, all these different things go ahead, and this little plant's trying to survive in it with minimal root system, drought susceptibility. That's when all of a sudden the site says, okay, plant, can you survive or not? And if you can't survive, more than 99% of them just plain die. And so in that opening, if you'd cut down that original parent tree, it would have 15,000 little baby seedlings could survive there, but they'd be reduced at this rate down to 150 during the first 3 years, and eventually only 1 or 2 would occupy the opening. That is the basis of mass selection breeding. If you want to learn more about mass selection breeding, I highly recommend reading the works of Luther Burbank. He's the guy who was born in Lancaster, Massachusetts, right down the road from me, one side of the hill from where I grew up. He has more plant varieties to his name still to this day than anyone ever in history, from the thornless cactus to the Shasta daisy. The Burbank russet baking potato, and on and on, and all kinds of plants he's got. The prune plum. This is a photograph underneath the tree on my farm. We call it grandmother maple. And this is what we're talking about. This is how nature plants little tree seedlings. If you counted them all these little slightly, bronzy type leaves are all little baby sugar maple seedlings. 99.999% of those are going to die. They are not fit to survive on this site. Although the selection pressures exist at all stages, a fully mature tree still has to withstand the frost and the cold and the dry and the hot. The selection pressures, the natural site characteristics, are the most effective on young seedlings, and in most species they will eliminate all the seedlings that are not adapted to their immediate environment. And this is one of the phrases, I don't say this to my trees, but this is the rule on my farm. Die now. You don't belong here. I can't waste time. Think about the difference between think about the difference between a craftsman apple orchardist, and I used to live on the same orchard with this one guy who's a craftsman apple orchardist. He will dig a hole in the ground. He'll dig a $500 hole for a $50 tree. Make the most incredibly beautiful soil environment that tree could ever have. He puts that tree in place. Perfect weed control, perfect water, perfect pH, all the minerals that it needs, foliar sprays, this, that, and the other thing. He just planted a young tree in the ground. What's the percentage of young trees that survive in nature? Less than 0.1%. So why are you investing so much into one little tree? Let's do it the other way around. Let's do it the way nature does. Let's put a shirtload of trees in the ground. I said shirtload. This thick, just like this. And they will sort themselves out. They'll go through this phase. This is site initiation phase. Now they'll go through a stem reduction, a thinning phase, where only the ones that are really, really site adapted will come to dominate that site. And look at this tree. Why is it that they're all the same age? And this one is somehow grown that tall. Now, if tall, straight oak timber is what we're selecting for, maybe that's what we want. However, right over here, look at this little stunty, runty little one. Well, that's getting selected against in this environment, but what if this was one of the ones that had a flower right off the bat? We want to be here and see which ones have precociousness, this young little plant flowering right away. That's a trait that we want, because this will allow us to rapidly accelerate producing new varieties as fast as possible. So this is the time. This is the time when to plant them really, really, really thick together, because a tree like this, even if it's only $5 per tree, 5, 10, 15, 20, 25, 30, there's $30 worth of trees in this area. These are at 3-foot centers, so 5 times 3, that's only 15 feet. If you were going to plant chestnut orchard, these are chestnuts, on the recommendations from Michigan State University, you'd have one here and one down there at the next one. 2 plants. You put this little seedling in the ground. If it's one out of the 99.999 that suck, you're stuck with a tree that's horrible, and you're going to spend the rest of your life trying to keep the doggone thing alive. Let's cram them in and find those genetic variations that are going to be successful on our site. Because how can we ever expect to find superior plants unless as a normal course of doing our agriculture, we do the massive fitness screening that nature does. Look how close these ones are. These were put in, I believe, in 1 foot apart. This was at the University of Illinois Woody Perennial Polyculture Research Site in Urbana, Illinois. I had the good fortune to plant all kinds of trees in one of the first corn variety trials that was funded by a land-grant university in the USA. Sexual reproduction is where what it's all about. This is how we get variation between individuals. This is how we get change in a community. This is how we get change within a population. And adaptive change is much faster in a sexual system, where we're constantly generating, we're rolling those dice, as fast as we can, as fast as we can. We're going to let all these plants cross-pollinate with one another willy-nilly. The ones that we don't want to pollinate, we'll cut them down, so they're not going to cast any pollen into our good trees. We'll keep our superior plants, selecting for the traits that we want. What traits that we want, that's our choice of what we choose. Lots of flowers, lots of reproductions, lots of seed, lots of mortality. That's the way nature does it, and we're okay with that. It's no big deal. Now, that's the sexual reproduction pathway. That's what really puts a lot of the variation in there. That's where we're going to find a lot of our magic plants. The apomixis, however, is a different type of reproduction. Typically, it's not considered the production of adventitious roots from a stem, like this one right here. This tree laid down onto the ground once upon a time when the sand was here, and it sent down roots. That's a vegetative reproduction, like taking cuttings. That's in some circles. That's not considered, apomixis. It is, however, the production of seed without pollination. There are hazelnuts that we have on our farm. I've noticed it in hazelnut plants that have a heavier infestation of big bud mite. They'll tend to put out some, apomictic seeds. You'll just see this. It looks like a pollen catkin, but then it made a seed instead of casting pollen. Some plants only reproduce by asexual reproduction. Others just do it occasionally. Sprouting from the stump or from the roots, falling fire or cutting. One of the ways to propagate trees, plants that do this is to take the roots and slice them apart, mow the plant down to the ground, so it'll stimulate this kind of sprouting. Then you run through and crisscross it with your subsoiler, and you just hack the roots up in a million little pieces. You pull the roots out and you plant them in. It's a great way to rejuvenate raspberries and blackberries, that sort of thing. And these are some more, purely apomictic plants. Egyptian walking onions, any of the hard-necked garlics, where they don't have a seed, but they have a little plantlet, a baby plant right up on top. A lot of tropical plants, house plants that you'll see will have these little baby plantlets form out in the leaf margin. A lot of blackberries and raspberries, they don't require pollination to produce a fruit. Mulberries don't require pollination to produce fruit. And one of the classic delicious plants right here that produces little baby plants sticking right off the stem out of the top is the pineapple. Sometimes they make little baby pineapples around here. Other times they'll sit one right up on top. And then I've even seen a pineapple plant like this with the fruit that we're going to eat. Whoops, go back. The fruit that we're going to eat is sitting right here. And then sometimes it sends out this little stem with another little fruit growing on top of that. So that's an asexual type of reproduction. Now, gene ecology is the study of the genetic variation in this plant species from an ecological viewpoint. So instead of looking at the phenotypical difference between these white spruce in Alaska, this white spruce in Minnesota, and that white spruce in Washington State, instead of looking at the appearance of it, it would be more understanding the genetics. Because, like I'd said earlier, if these guys, as a population, this place selects for plants that can survive super, super cold and, in this case, super acid soils, whereas this is a lot warmer. Temperate Olympic Peninsula is a lot warmer, a lot moister, so this is drier, this is colder, this is drier, this is hot. No organic matter. This could be almost boggy sometimes. Traits that will allow you to survive on a rock are the only ones that will allow this plant to survive. Anything that requires living in a bog goes away. Those traits just don't exist because they won't thrive on the rock. So the genetic makeup of this plant is more than likely radically different from the genetic makeup of this plant and the genetic makeup of this plant, but they're still all white spruce. They can still cross-pollinate with one another. And think about that. We have super cold tolerant plants. That could be a useful trait for us somewhere. We have a super hot and dry tolerant plant. That could be useful traits. We can combine the two. We can move traits back and forth. We can use this plasticity, not just the phenotypic plasticity, but the genotypic plasticity, to work with plants and change them through time. So here's just an example of chestnuts down the bottom. If you look at the range of American chestnut, how it would have ranged all the way up from wet 70 inches of rain, coastal fogs, super cold winters, 40 below zero, all the way down to Mississippi, Louisiana, where it's obviously a lot warmer. And here on the left, that's El Castaño de Cento Cavalli. That's the largest diameter trunk of a tree. This is basically the size of a city block. It's a huge tree. It's growing in the rock on the side of Mount Etna in Sicily. It's a Mediterranean climate. There are chestnuts that are almost tropical. And so if we got stuff that's almost Arctic down to almost tropical, there's different genes in this than there is in this if it was grown up in coastal Maine. So that's the study of the genetics of those trees. Now, this is where we start to get practical, of course. We are forest genecologists. We're going to collect seeds from target populations. What I did when I started with chestnuts, I got seed from Canada. I got it from the West Coast. I got it from California. I got it from Louisiana. I got some from China. I got from New York, from Minnesota, Michigan, all over the country. And when I'd bring it in, I'd put it in as a block so they'd be near each other and I could observe their behavior. Every time I brought in like a subset of these plants or seeds, that's a provenance. And when I'm testing these various different seedlings, that's a provenance test or seed source test. And it determines if there's any significant differences among that population, or between populations. Another thing that it does, if you then treat it with my classic patented sheer total utter neglect and abuse, we'll throw that one in there, you now are going to select against the characteristics that don't thrive on your site fast. I want them to die young. If they're going to die at all, get it over with. Don't waste my time reproducing and passing genes down that aren't going to be helpful long term. So these provenance testing is where we're going to start. We're going to grab a huge bunch of different genetics from a bunch of different places, put it on our property. Our first phase is this variety trials. We're doing provenance tests. It doesn't exactly tell us why a plant failed to thrive or what caused the differences between the plants. And I don't necessarily care. If it's dead, it's dead. I'm not interested. If it's not thriving, I don't care why. Just amend the soil to do better. It's like, no, screw that. If it's not doing well, it's not doing well. I'm not interested. We can try to infer what's causing these differences, but you can get in trouble there because oftentimes you can set up testing questions that you can prove over and over again something because of the evidence. You can prove over and over again your hypothesis of what caused the difference, but you can be wrong. Let's take a rock and drop a rock to the ground, and you can say, oh, the earth sucks. And you can do that test over and over and over again, almost willing to bet you a nickel that that rock's going to go down to the ground. So you prove your hypothesis over and over again that the earth sucks. And that might not be the mechanism that caused the difference or the survival difference. These are some locations. Once I started the provenance tests at my farm. Now here's the ones that are doing really, really well at my place. I start to get enough seed. I'm doing my own nursery beds. Once I get to a certain size and a scale, I want someone to produce lots of these numbers because if we're going to have a lot of plants and treat them abusively and don't feel bad if some of them die, we want to have some professionals raising them for it. So I contract out after a number of years, have people grow my seed for me. I supply them with the seed free of charge. They grow my plants. I pay them for the service to grow my plants, and then I plant them out. And this is one in a hotter zone, and this is one in a cooler zone. Not absolute cold, but colder degree days during the season. So there's slightly different soil conditions, different growing conditions during the year. But these are the kinds of numbers that we want to look at if we're going to be really screening for the genetic material that's going to work in these systems. So now populations that differ in one or more traits eventually can be called races. And so let's go back to pinon here. These pinons way up here in Utah. And what's north of Utah? Is that Montana up there, I think? Oops, my cursor went away. So you take pinyon pine populations up here versus pinyon pine population down in southern New Mexico, almost in Mexico itself. They've they have been so isolated from one another, they've not cross-pollinated with one another for a long, long period of time. They could be developing very distinct traits that are very typical of only that small little gene pool. They're almost separate gene pools. Those could be races. So once we go through the provenance trials, now we start to select for the plants that are doing really, really well on our site. Now we're beginning the process of narrowing traits down into races. And we will develop a land race of, pinyon pine, or of, sugar pine or Italian pine, whatever the species are. After we go through this huge combination of genetics from all over the place, then we'll start to do our selection on site, and eventually we'll develop a specific land race that has very specific characteristics of what makes it notable from others. Then when somebody else wants to start at, say, if I'm here in Wisconsin and somebody wants to start doing this over in British Columbia, okay, well, they'll start by grabbing material from all over the place, including my genetic material, and now they can add to their gene pool with material that I've already selected for certain characteristics. They will know about the characteristics, so we pay attention to that. So a lot of genetic differences happen when populations are grown in significantly different conditions. We've talked about that quite a bit already. Some of the things that really matter to us a lot are when they first, the buds burst first in the springtime. Oftentimes it's temperature related. Sometimes it's more photoperiod related. When they actually flower during the time of the year, and even between individual plants, there'll be like these family lines of plants that flower early in the season, these flower middle of the season, and yet the whole season happens to be, May or June or whatever. Well, then when it sets buds in the summertime, when it ripens the seed, when it stops elongating, all these are critical physiological changes that can have major differences between populations, and we'll want to be paying attention at least to these in our breeding and our selection. And like I said, they may have different triggers. They can be photoperiod, cessation of growth, for example. The classic, I think I've mentioned it before too, is when people like the big box retailers in Anchorage will look at the USDA zone map and see that, well, Anchorage doesn't get all that cold. We can do USDA growing zone 4 plants. And so they get plants from Missouri or whatever. They bring them up to Anchorage. Well, it's still warm enough during the days in September or so, so that the plants don't shut down. Well, you've got to be really photoperiod sensitive the further north you go, that as soon as the photoperiod goes past a certain time, you've got to shut down, harden off your growth, set those buds, and just go dormant before the temperatures tell you to go dormant, because if you don't, you're in for a big surprise, and you won't be very reproductively successful in the future. And then the populations will differentiate if the site conditions or disturbance regimes radically different. And this is where our selection comes in. Now think about, let's go use our example of going to Alaska and Missouri again. Let's take a willow let's do something different. Let's use a cherry tree from Alaska. It's settled in pretty well. This happens to be a willow in the picture on the left. Well, let's take a cherry tree from Alaska and then move it down to Missouri. Well, what might happen is the Alaskan tree has to start opening its buds and flowering early in the season when the temperature hits a certain number of degree days. So it may open up too early. It might open up midwinter down in Missouri. Then a frost comes by and kills all the flowers or kills all the leaves. And on the flip side, if we take something from too far south and we move it up, and if it's temperature sensitive, it may never move. It may just sit there. And this has actually happened a number of different times with nursery shipments up in Alaska, is you get nursery stock up that just never wakes up. It just never wakes up because it's too cold. There was just not enough heating degree days to get the plant to wake up. These are two examples. I use this. This is up in Alaska. This is willows. And notice I got pussy willows on it already, and the ground's covered with snow. They got to get started so early it's not funny if they want to finish, setting seed and all that. And these are willows. This is just taken in October. This is down in Louisiana. Same species of black willow, radically different. There's radically different genetics going on in there, radically different phenotypic response, different triggers that'll tell it when to get started, when to grow, when to shut down, when to set seed, etc. Ecologists use the term niche in an attempt to express in one word where and how a species is genetically adapted to persist. And there's spatial niches. Sugar maple, for example, is usually an upland species, well-drained soil, at least decently well-drained soil, whereas red maple can survive in swamps. And we'll talk about red maple a little bit more later on. It's one of the most widely adaptive species in North and Central America is the red maple. So there's the niche in space, up high in a landscape, down in a wet spot, dry, wet. Also in time, there's different niches in time. And if you have, say, a blowdown up in the boundary waters, so I took pictures up there this summer, and all these trees fall over, there's all these exposed soil, the light wind-blown seed from birches blows in, gets sprouted. Well, hemlock, eastern hemlock, has a lot of light wind-blown seed as well, and it goes in there and sprouts at the same time. The birch grows really, really fast, really comes to colonize that site early on, and then the hemlock grows slower, it takes its time, it can tolerate the shade, it can tolerate a little bit of extra dry because the birch has taken all of its moisture, and all that hemlock has to do is outlive the birch and eventually will dominate the site. Same with balsam fir. This is a red spruce up in Maine, red spruce, balsam fir, eastern hemlock, sugar maple for that matter, sugar maple and beech landing in a situation like this. All they got to do is outlive that early successional tree and they're going to dominate the site. So think about what we can do there with different niches in space on our property and different niches in time. If we set up the whole successional pathway early on, now we can remove, and we know what to remove when. We know that these are going to live 60 to 120 years and then die. Well, let's remove those first, and then these will dominate. So if we remove all the birch, it will be dominated obviously by these evergreens. If we remove all the evergreens, the birch is only going to last another 30 or 40 years and start to rot and fall apart, and maybe it'll reproduce, but maybe not, most likely not, because it needs a massive disturbance. Then if we clear cut it, now we've started over with a massive disturbance. So 3 different techniques here. We can remove the evergreens, and that will result in a not so strong population of the birch in the future. We can remove the birch, and it'll be a very strong population of the evergreens underneath it, or we can move all of them. 3 different management techniques, 3 different responses in the plant community below. So the gene flow through the generations keeps the populations from differentiating. And if you look at a species, like they kind of stick together and they're distinct. It is fascinating that there is some slight changes in varieties from land races, narrowly adapted to widely adapted plants within the population. But isn't it interesting that we have yet to observe an actual speciation where this species turns into something totally, completely different? So there's a certain cohesiveness to the gene pool, and that's restricted by number of breeding individuals, difference in flowering times, factors that control the viability of the embryo, the success of seedling establishment, and the isolation of breeding age individuals. You can get populations that are isolated, and they will become differentiated from another population if they're nowhere near each other. And that's the Alaska versus the Louisiana comparison. There will be different genes going on. Whereas if the population is all intact, there'll be gene flow between it, and you won't have traits disappearing that might be valuable traits to have. And some of those valuable traits might be disease resistance, pest resistance. Now, hybridization is accelerated in species or in regions where there's frequent disturbance. And there's the species that are adapted to frequent disturbance, like birches and poplars, aspens, willows. They cross-pollinate with one another like crazy, and there's all kinds of different hybrids between the two of them. I believe this is quaking aspen, quaking aspen and big-tooth aspen, and everything that's like right in here, it's a combination of the two. You're going to have these combinations, these little hybrids between the two that you won't be able to figure out by the phenotypic appearance, and it doesn't really matter. Well, how we can use hybridization is we can use it to enrich the gene pool of a species by introgression. We're gonna introduce new genes to a gene pool to hopefully give it characteristics that will help it to survive. And the classic example here, of course, is American chestnut. This is a classic American chestnut, straight and tall, shade tolerant, grows for 4, 5, 6, 700 years, produces nuts every single year, lethally susceptible to chestnut blight that the Chinese chestnuts were resistant to. So the American Chestnut Foundation, acf.org, took Chinese chestnut, crossed it with American chestnut, saved the seed from American chestnut, and planted it, the back cross. And then they were all of intermediate susceptibility. Some were susceptible, some weren't. Well, then they crossed it again with the American, and then did it a second time. Then some of them were intermediate, and then they crossed it a third time, and then you end up with 1/16 of the population. And I'm not going to bother to go into the genetics on this. This is called the third backcross generation. They've introduced Chinese chestnut genes into an American chestnut, and it looks like an American chestnut, acts like an American chestnut, eats, sleeps, and drinks like an American chestnut, but it has, you know, just a handful of Chinese chestnut genetics in it. Now, another thing that we can play with a little bit is polyploidy, and this is something new that I learned in getting ready for this particular webinar. Is that birches are one of the most polyploided trees that are out there. When there is a pollen grain or a ovum, they have half the chromosomes that a actual whole plant would have, and that's the haploid generation, or the gametes. Once they're together, now I've got the zygote. It's diploid. We're all diploid. 23 chromosomes. We got, you know, one from mom, one from dad. There's 2 of each all the way across the board. Well, there are cases where then there are 3 chromosomes of each, 4 chromosomes of each, and yellow birch, as a matter of fact, is hexaploid, 6 sets of chromosomes. So there's, you know, where they all came from, I don't exactly know. And just in the last 10 years or so, there's been discovered on Lake Michigan, on the lakeshore in Lake Michigan, an octoploid birch tree. I don't even know if they've given it a name yet. Thing with polyploidy is it slows down differentiation, because if you want to try to get, if one of these genes codes for disease resistance, these 3 are susceptible. You can't get that disease resistance to show until you have resistance all the way across the board here. So getting the recombinations just right to make it completely resistant is a lot trickier in a polyploid organism. So all kinds of crazy things happen with polyploidy, and one of the things that I like to share with people who are considering playing with polyploidism is the colchicine crocus, Colchicum autumnale. It's the root of the colchicine crocus is totally poisonous, and if you take the bulb of the root and you squeeze the juice out of it with a garlic press, then you put seeds into it, it will kill 90%, if not 99% of the seeds that you put in there, but the ones that remain behind are now polyploid. Will those polyploid seeds grow, sprout, and propagate? We don't know until you actually plant them. So it's a little bit of a crapshoot, but it's has some interesting side effects that actually have some economic value to them, and probably one of the more economic crops where polyploidy has really come into play is in smoking herbal medicine. It is a plant called Purple Haze X Northern Lights. Purple Haze X Northern Lights was run through the polyploiding process using juice from the colchicine crocus, and what formerly was a little flower bud that looked like this, now creates these crazy buds that look like that. So if you're in the business of selling herbal medicine with crazy buds, this obviously had an economic advantage for you, because now you're able to sell more of them. What can it do with trees? I don't know. We're going to cover some more things quick. I want to burn through this session tonight so we don't have to go over it again, even though we are a little bit over time. Wayne blabbed early and ate into the time, so I'll just keep going. I'll have a few slides left. Fitness and flexibility. Fitness is adaptation to a site and certain conditions. Endemic plants are extremely site adapted, not very flexible. This is Plitvička Jezera in Croatia, where I went a year ago. Spectacularly beautiful place. Limestone bedrock with hypercalcium alkaline water coming through it. There's so much calcium in the water that it precipitates out and makes these amazing rock formations. And you've got some totally unique plants that only exist there. Another one of my favorite places, of course, is walking back to my homestead. If I go this way, all along here, there's 3 different mud volcano hot springs areas. And if you can imagine the hot springs with like scalding hot water, this is not comfortable to sit in the hot tub water. In the middle of 50 below zero winter, you've got plants growing all the way around the rim here that exist nowhere else on the planet. That's very adapted to that site, but it's not very flexible at all. It's very fit for those conditions, but not very adapted, not adaptable. Now, a cosmopolitan organism is widely adapted to so many different sites. That's like the red maple. It's like willows. This picture here, these are red maples up here. You know, less than 150, 200 miles from the Arctic Circle. And these are red maples down on the bayou in Louisiana where we were last fall. Now, inbreeding, apomixis, polyploidy, and a low rate of combination are uniformity promoting devices that favor fitness, but not necessarily the adaptability and the flexibility. Flexibility is favored by variability promoting mechanisms such as cross-pollination, precociousness, the likelihood that they'll create natural hybrids, precociousness, early reproduction, heavy seed bearing. These are traits that are very, very useful to us as farming. So to answer one of the questions, well, within what time frame, this is why the first thing that I look for and select for is precociousness. If you aren't producing seed early and young, I'm not interested. And if you're not going to produce a lot of seed, I'm not interested. And that's how we get a lot of recombination, is by reproducing and flowering over and over and over again. We even have some chestnuts that produce 2 sets of flowers in the summertime. That's a lot of recombination. Now, this woody cropping system is a result of 20 years of provenance testing. It managed as well as natural selection. It's disturbed according to the historic trends and as well as to its species adaptations. The crop production is based on the population fitness, not the specific characteristics of one little plant. It's this whole population of plants as a unit, as a system, is what's fit, not one particular plant. And then through the managed and natural selection pressure, we optimize yields to the site conditions. This whole system is resilient. It is part of reality. It's been through tornadoes. It's been through fire. It's been through hailstones the size of a dinner plate. It's been through 50 below zero. It's been to 116 3 different times in the summertime. I wonder why 116 and not any higher than that. That's been 3 different times. This system is a part of reality. It does not need to be protected from reality. Every disease of these different species exists on this farm in high population levels. It's the system that is resilient, not the individual plants that are resilient. Whereas this can be certified organic, perennial, part of permaculture design, holistic and managed system. It's just based on a mere handful of genetically identical plants. It's all the same genus and species, Malus domestica, maybe 5 varieties here. It's maximized production of one particular yield, apples. But the ecological processes now have been externalized, and we now have to perform those functions. We have to perform the pest control. We have to perform the disease control. We have to perform the fertility management instead of allowing the system to actually take care of itself using natural processes without our interference. This quote-unquote orchard is radically different. It's much more resilient. It'll persist over time. It's an ecologically regenerative system in that it is site adapted and it's able to regenerate and maintain itself. You know you're doing something right when you start having more apple trees and chestnut trees and hazelnut trees and pine nut trees just showing up all over the place because your system is working, you're doing something right, and you're having really fit survivors. One of my favorite ones that I like to see all over the place, I never know where I'm going to find the next one, are Nanking cherry. It's a bush cherry, little sweet cherries, and you never know where a bird took that seed and dropped it and pooped it out. It'll survive on my place because it's fit and it's adaptive. Genotypic plasticity, the plasticity of the genotype, of course, and the phenotype as well, combined with mass selection breeding, disturbance mimicry, complexity of the ecosystem. This optimizes the total site yield. That's one of my jobs as a system manager, is to manage all of this and keep it on track so that the species that have the traits that I want are the ones that remain behind, and the ones that I don't want become disturbed and become another source of income. See this tree right over here on the right? These are some of the first known Asian pears growing on a commercial scale in the state of Wisconsin. They are a result of seeds that a student of mine saved and planted and selected for and grafted, and then once they were successful at their place, we took them over to my place. And they withstand outstanding, amazingly cold temperatures, extremely cold temperatures. We also still have annual perennial herbaceous plants that are in there to interact with severe frequent disturbance. We're doing successional soil building with a succession of species, beginning with our simplest ones to, through our grassy phase, through our shrub phase, through our pioneer tree species, woody species phase, to our mid-successional phase and late successional phase as well. We're doing annual grains. We're doing silvopasture. These are the alley cropping, silvopasture, our agroforestry techniques. We're growing trees and branches, and we're chipping them right where they grow. There's ramial wood chips for fertilizer, for soil building. Obviously, the animals in the system. Are you guys starting to get this at all? Are you starting to get this? I'm going to close on that because we went over time, and the rest of these I do have a little tidbit on last summer. I spent some time up in the northern Italian Alps, and I've got some pictures of the villages that we went to up there. These abandoned villages, these old ruins. I can address that next time. It's pretty easy. We got plenty of time. So you're starting to get it. Good. At least Nick's starting to get it.
Thank you, man. You got several questions. You open for a few questions, Mark?
Oh, yeah. Yeah.
All right, cool. And Jian has been coming, and I believe he's in Australia, and he's been coming to a lot of sessions lately. I got to give you a funny one. Sam actually asked a question, and then later he said, I wish I would have been more patient because Mark already answered it. So,
okay.
So we're thinking on his. So Jennifer asks, and Jennifer's down in Arizona, by the way, Mark, and she's asked a couple questions here, but is there a white spruce that would live in very alkaline soil? I'm at 5,900 feet in northeast Arizona.
What you do is you go grab white spruce seedlings from as close to the similar climatic conditions and soil conditions that you can possibly get, and you plant a zillion of them. White spruce, it's one of those real cosmopolitan species. It has a lot of genetic tricks going on in there, and so I wouldn't be surprised if there was. I would try some nurseries maybe in Utah, high elevation Utah, because they have a lot of alkaline soils there. Yeah, I wouldn't. I'd just start there. Try to find nurseries near you with similar conditions, and buy seed if you can, if you want, or buy the cheapest seedlings in bulk and plant a lot of them.
Well, she has an associated question, so you probably already answered it, but I'm going to ask anyway. Any sources of genetically superior pinyon pine, Edulis, for the northeast Arizona? I have several on the property, but want to plant more.
Yeah, I don't know anybody yet who's really actively focusing on genetic improvement of pinyon pine. We're not even, as a nation, we're not utilizing pinyon pine the way we could, and we're not doing any breeding work on it, so it's up to us. So there you go. Get your eyes open and start looking around, and mark your trees. Look for trees that are growing vigorously, that produce lots of cones, lots of nuts, that don't have a lot of pest problems, that don't appear to be infected with diseases, and take it from there.
You have a business, Jennifer, there,
There you go.
and we'll talk with you more about that. We have some other plans down the road for New Forest Farm, and we'll talk more about that later. Jen asks, and I might even know the answer to this, but I'm going to let you answer it. Can you bring seeds from other countries into the U.S.? And Jen says in Australia we cannot do that. So.
Yes, you can. There's certain processes that you have to go through in order to do it, and it's laborious and time consuming. I actually have 3 different plant smuggling convictions, and so I no longer do it informally. I only do it formally because what is wanted is to prevent disease, bringing in disease and stuff like that. So I know that when I've shipped seed to other countries, it's taken probably 2 months to go through the whole entire process of phytosanitary. Costs a couple $300 to do it. And then
Awesome.
I also see the same person asking a question about what time frame that chestnut seeds can be kept to be still viable. And here's
Yep.
how I've operated with seeds and plants. I'm thinking that whether this planet was designed, evolved, adapted, or sprang out of nothing instantaneously, whatever, there's a reason why fruit becomes ripe and or nuts become ripe. It's that time of year that when you take that fruit and you cut it open, put the seed in the ground. Because probably in the ground is where the right conditions are for that seed to sprout. Otherwise, why would a seed have been invented? So when cherries are ripe in July, that's when the seed goes in the ground. When the pears are ready in late August into September, that's when the seed goes into the ground. When the chestnuts are falling off the tree in October, that's when the seeds go into the ground. I mean, all of the stuff, you can go online and get books galore and learn how to stratify seed properly so they'll sprout. It's like, well, give me a break. Put it in the ground. When it comes off the tree, put it in the ground. It works. How long is it viable? Well, how long does it take to get off of the tree and put it in the ground? That's all the time that you need. The seeds will be viable. That's another reason why, like in nature, there may be only 1/10% of the trees actually turn into a seedling that actually survives. It's like a merciless task. So get a whole shirtload of seeds, put them in the ground. Okay, now the stupid.
You're seeing
answer.
the question.
Now
Actually, you see Sam's? You see
What?
Sam's question? You see Sam's right after Jen's there?
Which one? With the
Yes.
well, with the selecting for what the question of do I hold selecting for just 1 or 2 traits per generation? That's a question that's coming from a understanding of classical genetics, in that the number of traits, in order to change one trait, you're going to have to have a whole bunch of accessions, different rolls of the genetic dice each generation just to change one trait. So to change 2, you don't just double the numbers of trees that you need in order to get those changes. It's geometric through time, and so it's insanity to try to change more than one trait at a time. I'm not talking about classic genetics. I'm talking about managing a herd, and I'm going to manage I'm selecting for my chestnuts cold hardiness, hyper precocity, heavy yielding, pest and disease resistance. 5 different traits all at once. And then, do I all of a sudden get all of those 5 traits changed in one generation? Of course not. I'm managing a herd through time. And as long as I've got this hyper precociousness in the gene pool, I've got the opportunity, if I want to, make some controlled crosses using a hyperactive, female, and I know if I just pollinate her with somebody else, that seed's likely to have, a flower the next spring. Can I change all 5 in one generation? Of course not. I'm talking about changing the face of agriculture and to now start mimicking natural processes with mass selection, guided mass selection. We're going to select for as many traits as you want if you want. And the numbers go through the roof. That's why we plant so many trees. That's why we plant so many trees.
Do you see the question there? I think pretty cool question or comment from Andrew coming down just a little way from there, Mark.
How many is a zillion? Yes, it is. How numbers of trees per acreage would I initially plant of fruit trees? What type of fruit trees? What size is your site? What are your resources? If you're going to be doing fruit trees, most fruit trees that I know of are sold as grafted clones. So they're clones. What I did is I bought rootstock, seedling rootstock of Antonovka. It's a real cold, hardy apple. And then we know, though, that Antonovka, most of those rootstocks probably will be junk. So what? You let one of the branch of the root grow, and then you graft a variety on top of it. So in my first apple plantings, I think I did about 700 or 800 per acre. I gave them a little space, and it wasn't. I hadn't quite fully drank the Kool-Aid of high density at that point in time. And once I got enough apples, I didn't need to plant anymore, anyways. But with the chestnuts and hazelnuts, the high density, I'm going high density all the way, and with the pine nuts as well. Right now, speaking of pine nuts, I know I'm not going to pitch or anything, right? Go to forestag.com. One of the things we're talking about seed, put it in the ground. Korean pine is one of the biggest pains in the butt species to sprout. Sometimes it sprouts, sometimes it doesn't. And like this past year, it's like the seeds went in the ground and you couldn't keep them from sprouting. There's like two trees came out of every seed, it seemed like. So we have a real lot of the Korean pine available yet this spring, as well as the hazelnut, selected hazelnut seedlings. So a zillion, aiming for both economic and genetic viability, it's a crapshoot. It's totally variable, and there's too many variables to just give a quick answer to a quick question like that. And
so I'm answering both of Mary's questions here, Mark. You can look at them, but I think that the first one we can answer separately, sort of in more generic form, and then the second one, she asked about a specific site on the plan for the assignment, and I answered it the same way you did last week, which is you do not have to have a specific site, but and for an area would be really good. But I answered it saying if you did it for your specific site, then as we review it and critique it and such, it might add more specific value to you down the road. So that's kind of my answer.
Yeah, but you don't need to own a piece of property in order to do the exercise. It's just for your particular region or your chosen region. If you want to like pick coastal Chile at 45-degree latitude, that's perfect. Go for it. But I want it to be legit. I want it to be based on ecological realities, because if it's not observably ecological, here it is. This is an actual plant community observed in this particular place. If it doesn't have any, peer reviewable science behind it, not interested.
Yeah, and I think part of the reasons Mark's saying that, Mary, is that we want to get as broad of a diversity of sites as we can all over the world. And we know we're not going to get all of those this time around, but if we keep doing this, we'll have an amazing database that will be helpful for everybody. And Jennifer, the homework side is, again, we're hoping to have that done by the time Mark finishes. When he first gave the assignment, there was going to be like 8 weeks left. Maybe he can comment quickly whether he thinks he's still about 6 or so, but that's the idea. The timing is for when this series and this course finishes.
Yeah, I think 5 or 6. I haven't looked at my outline, but I do know that next week we'll either be doing plant structure and physiology is probably one of them that's coming up next. Because as you see, what we did is we went through the broad stuff. Now we're getting more into specific details. And actually one that I want to touch on because it's relevant to where discussion is these days, and that's the term regeneration. Regeneration has a very specific ecological scientific meaning that's got a 100-year track record of research and PhDs and colleges and universities all around the world have agreed that this is what regeneration means. Well, now it's kind of getting buzzwordy and jargony. Well, regenerative agriculture. Well, the term as it's being used quite commonly isn't being used the same way as ecologists and foresters and natural resource managers, range managers would use regeneration. And so I want to give a presentation, next week or the week after. I'll have to look at my list again on regeneration ecology. It's a very, and I'm not going to do it to address regenerative agriculture directly. I'm doing this as an ecologist. This is the way ecologists view regeneration, and so that's
Super.
and talk about regeneration ecology. And,
so let's make this the last question for the evening. We've got about an hour and a half. Alyssa asks, are those tubes in the picture tree shelters? I think it's cool to end on because last week, remember, we ended on some pigs in the picture. So are those tubes in the picture tree shelters, or do you ever use tree shelters? And this is Alyssa asking.
Well, I've used tree shelters, absolutely, and in this particular context, they're perfect because if there's after we've gone into the system here and we've thinned out the underbrush, we acted as if we were a surface fire, except that we used a forestry mulcher, ground it all up, got the grass going. Where there was a big gap in the canopy, would plant another tree and would protect it with these tree shelters. I think it's treeprotection.org or treeprotection.com is the Southwest Badger Resource Conservation Development Council. They sell tree shelters as a fundraiser. Now they get the tree shelters from the company. The company donates it to the nonprofit as a tax write-off. Well, then the nonprofit sells it. They got them for free, so then they sell it at about the same price as the company does. But it all goes towards doing conservation, tree planting, riparian buffers and education and stuff like that. So I'd prefer to get them from the Southwest Badger Resource Conservation Development Council. They'll ship them anywhere around around the country. And there's been all kinds of research done on the color of the tree tubes. The colors that are used most commonly foster stem elongation. It's it's like a pink peach type color. All right, thank you everybody for being here. We'll see you next week.
Thank you, man. Talk to you soon, man.
Good night.
Bye, everybody.